So children, we have learned about prophase 1 in our previous part. Prophase 1 being the longest phase is divided into 5 different stages or 5 sub-stages. That is leptotene, zygotene, pachytene, diplotene and dikinesis. Now this prophase 1 is then followed by metaphase 1. In this metaphase 1, what happens? The centrioles which are there inside the cell, those centrioles, they move towards the opposite poles. As you can see in the diagram, the centrioles, they have moved towards their opposite poles. Now the spindle fibers which are there, which come out from the centrioles, the spindle fibers, they become well developed. They become more prominent and well developed. The chromosomes which are there, or the non-sister chromatids which are there, those chromosomes, they condensed even more, and as they condense, they become thick. They become more thick and they become more prominent. They become more visible, and that's why this metaphase 1 is said to be the best stage to study the chromosomes. After that, what happens? The chromosomes which are already attached to each other by the tip. These chromosomes here in the metaphase 1 also, they are attached to each other by the tip, just like they were attached in the dikinesis phase. Those chromosomes, they are attached by their tip, but due to the repulsive forces, due to the repulsion in between these non-sister chromatids, they are ready to separate from each other. So in metaphase 1, what happens? The centrioles, they move towards the opposite poles. They have moved towards the opposite poles. The spindle fibers which connect the centrioles, the spindle fibers become well developed and more prominent. The chromosomes which are there, they become more condensed and they become more thick, and they become more prominent or more visible. So that's why, as they are visible properly, clearly, this stage or this phase, metaphase 1, is said to be the best stage to study about chromosomes, to study the chromosomes. And as these chromosomes are attached to each other by the tip, due to the repulsive forces in between these non-sister chromatids, in between these chromosomes, there are repulsive forces. As the genetic material gets exchanged, after that these chromosomes, these non-sister chromatids, they repel each other, right? So here those repulsive forces, they act, and these chromosomes are ready to separate from each other. Then the metaphase 1 is followed by anaphase 1. In anaphase 1, the dysfunction of chromosomes takes place. That is, the homologous chromosomes are pulled away from each other and are carried towards the opposite poles, and the non-sister chromatids or the homologous chromosomes, they get separated from each other and they move towards the opposite pole. They get pulled towards the opposite pole. As you can see in the diagram, these non-sister chromatids or these homologous chromosomes, they have separated. That means here the dysfunction has taken place. They get separated from each other. This separation is called as dysfunction of chromosomes. They get separated from each other and they are pulled at the opposite poles by the chromosomal fibers. The chromosomal fibers which are present there, they pull those chromosomes towards the opposite poles. Half number of chromosomes start moving towards the opposite poles. Half of the number of chromosomes, they start moving towards the opposite pole. Now I have taken here 2 chromosomes. So I have drawn here 2 chromosomes. So one will move at each pole. If there are 16 chromosomes, so 8 will move at each pole. 8 here on this pole and 8 here on this pole. If there are 20 chromosomes, 10 will move towards each pole. So half of the number of chromosomes get pulled towards each of the pole. The chromosomal fibers pull the chromosomes at opposite pole. As I said, there are chromosomal fibers which pull those chromosomes towards the opposite poles. They pull these chromosomes towards the opposite poles. The chromosome number becomes half over here in anaphase 1. Anaphase 1 is the most important stage in meiosis 1, because here the actual reduction of chromosome takes place. Actual division of chromosome takes place. Now, as I said, the dysfunctioning of chromosome takes place. That is, the chromosome gets separated from each other. Now, they are pulled towards the opposite poles. These chromosomes, they are pulled towards the opposite poles. one on, as I have taken 2 chromosomes, so one on each pole. So this is where the number of chromosome becomes half. One on this pole and one on this pole. The non-dysfunction of chromosomes leads to the chromosomal disorder. Now, if this separation of chromosome did not happen, if this separation of chromosome doesn't happen, at that time what happens? This non-dysfunction of chromosome, non-separation of chromosome, it leads towards the chromosomal disorder. The person may suffer, or the cell may have chromosomal disorder due to the non-dysfunction of chromosomes. So, in Anaphase 1, the actual reduction takes place. That is, the separation of chromosomes, or the chromosome number becomes half. Here, the non-sister chromatids, they get separated from each other. Now, in this meiosis 1, the sister chromatids do not get separated. Now, as we know, what are sister chromatids? These, suppose this is the chromosome, they are attached to each other by a point called as centromere. They are attached to each other by the point called as centromere. This one strand of chromosome and this other strand of chromosome, they are said to be the sister chromatids. Now, as here there are two chromosomes, this one strand and this other strand will be sisters to each other. But this chromosome and this chromosome will not be a sister to each other. So, they are called as non-sister chromatids. So, here the non-sister chromatids, they will get separated from each other as they were attached with their tip, as they were attached by their tip. Now, here in Anaphase 1, they will get separated from each other due to the repulsive forces. Now, as they get separated from each other, that is called as dysfunction of chromosome. The separation of chromosomes, the separation of non-sister chromatids, or you can say the homologous chromosomes. So, that is called as dysfunction of chromosomes. Now, as they get dysfunctioned, they get pulled towards the opposite poles by the chromosomal fibers which are there. So, these chromosomes, the non-sister chromatids or the homologous chromosomes, they get pulled towards the opposite pole. For example, I have taken here 2 chromosomes. So, one will get pulled at each pole. If there are 16 chromosomes, so 8 will get pulled at each pole. 8 at this pole and 8 at this pole. So, here in Anaphase 1, the actual reduction takes place. The chromosome number is actually divided into half. Now, the Anaphase 1 is followed by Telophase 1. In Telophase 1, the nuclear membrane and nucleolus reappears. As you can see in the diagram, the nuclear membrane and the nucleolus reappears. It starts appearing again. The spindle fibers which were there, they get disappeared. The spindle fibers which were there in the Anaphase, they get disappeared over here. The haploid number of chromosomes after reaching to their opposite poles or to their respective poles, they become uncoiled and elongated to form chromatin material. Now, as you can see, the chromosomes, in the beginning which were diploid, they got divided into haploid, and these haploid chromosomes, after they reach to their respective poles, they uncoil themselves. Now, as I said, further they condense and they coil together to form the chromosomes, right? So now here what happens? They uncoil themselves and they form chromatin material. They uncoil themselves to form the chromatin material. Thus, here in the telophase 1, what happens? 2 new daughter nuclei are born, or 2 new daughter nuclei are produced. Now, this is the karyokinesis process ends. As we know, karyokinesis process, that is division of nucleus. So, here the nucleus gets divided into 2. So, 2 new daughter nuclei are born or formed. So, here the karyokinesis process ends with the telophase 1. In telophase 1, the chromosomes or the haploid chromosomes which were there, they move towards their opposite poles, and as they have moved towards their respective poles, they start uncoiling themselves. They uncoil and they elongate. They become long again to form a chromatin material. the nuclear membrane and the nucleolus, they again appear, or they reappear, they start appearing again, and thus the two new daughter nuclei are formed or born or produced. So here with telophase 1, the karyokinesis 1 process ends. Then the telophase 1 is followed by cytokinesis 1. Cytokinesis, as we know, that it is the division of cytoplasm. The cytoplasm gets divided into two new daughter cells. Now here, as it is an animal cell, here the cytoplasm will constrict in the middle. As it is an animal cell, the furrow formation or constriction of cytoplasm will take place. And as the cytoplasm starts constricting more and more, this will result in the formation of two new daughter cells, and the daughter cells which are formed over here, these two new daughter cells will be haploid daughter cells. These haploid daughter cells will then undergo the process of meiosis too. So here in cytokinesis, the cell or the cytoplasm of the cell starts getting constricted. It becomes constricted, and eventually it divides into two new haploid daughter cells. As you can see here, two new haploid daughter cells are formed, and these haploid daughter cells, then they will undergo the process of meiosis